All right. So just a reminder before we begin, just ask that everyone please mute your mic and turn off your camera until the end of the talk. Just save a bit of bandwidth. And at the end of the talk, if you have a question, please just indicate so in the chat. And then at that point you can unmute your mic and turn on your camera and ask a question. So before we begin, we would like to respectfully acknowledge the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk people. And we also recognize the Inuit of Nunatsiavut and Nunatukavut and the Innu of Nitassinan and their ancestors as the original people of Labrador. We strive for respectful and equitable partnerships with all people of this province as we work towards true reconciliation and honor this beautiful land together. So thanks everyone for joining us for the last seminar of the 2020-2021 seminar series. And today I have the pleasure of introducing Dr. Duncan MacKenzie. So Duncan has a PhD in zoology from the University of California, Berkeley, and did a postdoc at the University of Alberta. And he is now an associate professor at Texas A&M, where he studies comparative endocrinology and thyroid function. And today he's going to be talking to us about some of his research on the red drum fish and thyroid hormones. So welcome, Duncan, and you can go ahead and start sharing and take it away.
All right, thank you. Let me go ahead and start this up. Really pleased to be invited to give this talk today. I'm always very pleased to be invited by graduate students. I'm also kind of pleased that I don't have to travel to St. John's, not because it's somewhere I don't want to go. I have actually been to St. John's. I was there in 1987 for the International Symposium of Fish Reproductive Biology. And I learned something on that trip. When you fly into San Francisco and it's foggy, they close the airport. But when you fly into St. John's, it's such a commitment that regardless of how foggy it is, you land that plane in zero visibility. And it was an interesting experience, maybe somewhat stressful for the passengers, but it just proved how important it was to get there. It was worth it, and I really enjoyed my visit. It's really an honor to speak to this group. Memorial University has an outstanding history in fish endocrinology. When I was in graduate school, David Edler was one of the world's leaders in the productions of gonadotropin. When I was a postdoc, Larry Krim was someone we collaborated with extensively, working on fish reproductive endocrinology. And I've known Hélène Volkoff since she was a graduate student, and I have followed her career with great interest as she's developed one of the most productive nutritional neuroendocrinology labs in the world there in Memorial. So you have a fantastic legacy of fish endocrinology, and that was one reason I feel very honored to speak to this group. Also, in honor of that legacy, I'm going to kind of focus on classic old school fish endocrinology today. If you were looking for heat maps or RNA-seq data or deep dives into metagenomics, I'm sorry, you're going to be disappointed. I'm kind of at a point in my career now, just very close to retirement, where I'm getting much more retrospective and thinking a little bit about what's worked and what hasn't and what needs to be done in the future in fish endocrinology. I've also noticed from the literature that the Volkoff lab seems to be inching toward doing some thyroid work. And I may use this talk as a chance to both encourage them and perhaps warn them a little bit about moving in there. Just to give you some background on how I ended up here, let me kind of summarize quickly the odyssey that is my love-hate relationship with the fish thyroid gland. I started out, as you heard, at the University of California, Berkeley, working in the lab of Dr. Paul Licht. Dr. Licht and his collaborator at UC San Francisco, Harold Papkoff, were at the time asking what seems to be a fairly quaint question now, but was actually very important then, as to how many gonadotropins existed in the pituitary gland of non-mammalian vertebrates. And we're working to purify and characterize the biological activity of pituitary hormones. And the final answer was 2. In fact, pretty much what we expected from the mammalian studies. But at the time, they had a problem, and that is that the pituitary gland produces a collection of hormones, which are glycoprotein dimers, which are homologous and are very similar in structure. 3 famous hormones, LH, FSH, and TSH. And the Licht and Papkoff group were interested in LH and FSH in their control of reproduction. But the problem is the kind of purification techniques you used in those days. You would get contamination of those hormone preparations with the chemically similar TSH molecule, which also existed in the pituitary. So when I came in, my job was to sniff out the TSH and characterize it. And I spent much of my time at Berkeley kind of developing a variety of biological assays for evaluating pituitary function in stimulating the thyroid gland. And that involved creating a radioimmunoassay for the most common thyroid hormone, thyroxine, and then testing a variety of pituitary hormones. So let me get my little laser here. Testing a variety of pituitary hormones, including mammalian hormones and fish hormones and reptilian and amphibian hormones for their biological activity. And in the end, we were able to purify thyrotropin for the first time and develop an immunoassay for measuring it in reptiles. And my first exposure to fish came during this time when we took some of these hormones that we were preparing for reptilian, avian, and amphibian species and tested them out in the most convenient fish model that was available. This little guy, a long-jawed mudsucker, an estuarine goby from San Francisco Bay, which was available to us because Howard Burns' lab was using it to study osmoregulation. And when we used a lot of our mammalian hormones and other exotic hormones in the species, there were some pretty interesting questions, some unexpected results with fish, suggesting that there was a much closer interaction between the reproductive and thyroid axes in fish than we had seen distinctly expressed in other species and in a tetrapod species. So I developed kind of an interest in fish at this time. There wasn't gonna be a whole lot of room, I think, for doing work on reptilian endocrinology, and I was very fortunate to land a postdoctoral position in the laboratory of Dr. Dick Peter at the University of Alberta. Moving from Berkeley to Alberta was an interesting transition. But the great thing about the University of Alberta is that Dr. Peter had a system and a number of unique resources for studying the regulation of the pituitary, the hypothalamo-pituitary axis. In particular, he had been focusing on reproduction. But Dick had done, during his graduate work, when he was a graduate student with Aubrey Gordman at the University of Washington, done what, to me, was one of the most important experiments in the history of fish pituitary endocrinology. And it was a very simple experiment. He had simply taken the pituitary gland and translocated it away from the base of the hypothalamus. And when you move the pituitary gland in a mammal, the results are pretty obvious. The mammal stops reproducing. Reproduction and thyroid function diminish very dramatically when you break the connection between the hypothalamus and the pituitary. And when Dick did the same experiment in goldfish, he got exactly the opposite result, in which he saw an activation of both reproduction and thyroid function when he moved the pituitary gland, suggesting that the proximity to the hypothalamus was resulting in a tonic inhibition of these two axes. And therefore, in teleost fish, the regulation of reproduction and thyroid function by the brain might be exactly the opposite and of mammals. And Dick had done a lot of really nice development of techniques to evaluate this process. And one of the techniques I was particularly interested in, there's a goldfish right there. This is a lesioning needle that was being dropped into the goldfish's basal hypothalamus. We could go in and we could selectively eliminate certain neurons in the base of the hypothalamus. And in doing so, we could evaluate the impact of those hypothalamic nuclei on the regulation of both the thyroid and reproductive axes. And Dick had focused exclusively then at that time on the reproductive axis at Edmonton and had done some legendary work now proving that dopamine is a neurotransmitter that inhibits reproduction in goldfish. And I wanted to try the same thing to see if I could separate the reproductive axis and thyroid axis. We did these experiments, and fortunately I was able to confirm exactly what Dick had seen before. Essentially, what we found when we did this, and again, I was using a slightly different approach, but essentially what I was looking at was the results of lesioning of the basal hypothalamus on thyroid function in these organisms. And when you lesion the basal hypothalamus, it turns on the thyroid as assessed by the presence of thyroid hormone in the blood. Within 10 hours, we saw a spectacular increase after lesioning, suggesting again that we were destroying neurons that were somehow inhibiting the thyroid axis. And I remember Dick being very relieved when we got this result because it was the first time it had been confirmed in 20 years since he'd done his his graduate work. The other thing that I was able to do when I was at Alberta is I collaborated extensively with Dr. Norm Stacey. Norm was very interested in the regulation of spawning behavior and also used goldfish as a great model system to evaluate the interaction between the ovary and the brain regulating spawning behavior. But Norm also loved working outside, and he discovered a location about an hour out of Edmonton where there was a spawning run of another interesting organism, the white sucker. And we were able to do over 2.5 years field work evaluating, capturing these animals during their actual spawning activity, taking blood samples, and we're able to complete what I still think is, to this day, the most comprehensive descriptive study of changes in hormones, including reproductive hormones, thyroid hormones, adrenal hormones, growth hormone, during spawning in a wild species. So I kind of came out of that into experience with an appreciation both for the importance of laboratory models, but a desire to try and incorporate real fish in the wild into those studies. Now, the other great thing about Alberta was that Dick's lab was so productive in fish reproductive endocrinology that all the famous people came through there. In spite of the fact that it was 300 miles north of Montana, we had a stream of visitors from all over the world, Japan, United States, Canada, and Europe, who were in residence for some period of time. And one of the people who came and was in residence was Aubrey Gordman. Aubrey had been Dick's major professor at the University of Washington. Aubrey was the editor-in-chief of General and Comparative Endocrinology and also was essentially the founder of fish thyroid endocrinology when he started doing that work when he was a graduate student at Berkeley. And I had a chance, because of the fact that people stayed for a while, to have chats with Aubrey while he was there. And I remember one day asking him, so why did you stop working on thyroid function in fish? Because he at the time, he was working on the development of pituitary in embryos. And I remember to this day, I remember what Aubrey said when I asked him why he stopped working on fish thyroids. He said, because it was too boring. I expect that was Aubrey trying to be somewhat provocative, as he often was, but that's always stayed with me. But, you know, not necessarily the most inspiring thing to hear the most distinguished person in your field say about the career you're planning to make for yourself. So thus inspired, I left Edmonton and moved to Texas A&M University, where there was a great group in comparative endocrinology and the opportunity to work with a number of different fish species. So now that I get to A&M, I'm going to talk a little bit more about where we were, and I want to give you some background on just what the axis is. I know you're not all endocrinologists, but you've probably all seen this diagram before. This is what I refer to as the tyranny of the mammalian model. for comparative endocrinologists. If you open up any textbook or endocrinology textbook, physiology textbook, you're going to see this is how the thyroid gland is controlled. So at the top, you have the hypothalamus. The hypothalamus produces peptide releasing factors, and in mammals, the primary molecule is TRH, thyrotropin releasing hormone. That travels, in mammals at least, through a portal vasculature system to the adenohypophysis of the pituitary and stimulates the release of the molecule I was interested in, thyrotropin, a glycoprotein dimer, a large complex molecule, travels in the blood, binds to receptors on the thyroid gland. The thyroid gland accumulates all the iodine in your blood through a very complex series of biochemical steps. It manufactures its primary hormone, which is thyroxine, and secretes thyroxine into the circulation. And you can see right here that thyroxine has a unique structure. It's essentially an iodinated amino acid with 4 iodines located on the 2 rings. Thyroxine is the primary thyroid hormone that's in the blood, and it's going to be the signal that's driven into target cells to activate the important responses to thyroid hormone. Thyroxine diffuses out of the blood, probably facilitated by transport proteins into cells. And then the model you're going to see again in the textbooks is that there is an enzyme called outer ring deiodinase in target cells, which removes one of these outer ring iodines. And that's a critical step because that converts thyroxine with the 4 iodines to triiodothyronine, T3, with the 3 iodines. And in the process, it increases the affinity of this molecule for its nuclear receptors tenfold. And the model you'll see again is that T3 then binds to the nuclear receptors and it activates expression of specific thyroid hormone responsive proteins. In fact, it de-represses proteins which were being repressed by the unliganded receptor. And typically, the other thing that you'll see in a textbook is that thyroxine has negative feedback. And so what we see here is a system which we call a centrally powered system or a centrally driven system, in which the hypothalamus through the pituitary axis is driving thyroxine into the circulation. That thyroxine is deiodinated in target tissues and activates responses. And a situation called the pituitary thyrostat is established where T4 feeds back and inhibits the production of both TSH and TRH to maintain the homeostasis of the system. So this is, again, a pretty straightforward and centrally driven, simplistic model. And what you don't see in a lot of those textbooks are all the things that complicate this model. And that is all of the factors that function at peripheral levels, the peripheral control, to impact this system. What you don't see, for example, is there are alternate deiodination pathways. Outer ring deiodinase is an activating deiodinase, which takes T4 to the more active T3. But there is a second deiodinase called inner ring deiodinase, which converts T4 by removing an inner ring iodine. And remarkably enough, even though the formula, the chemical formula for inner ring deiodinated thyroxine is identical to the formula for outer ring deiodinated thyroxine, reverse T3 has no binding activity for receptors whatsoever. So this is a catabolic pathway which cells can use to basically say, I don't want to see T4, get rid of it. And they can even use inner ring deiodinase to remove the active hormone from cells. So if cells turn on inner ring deiodinase, they effectively can defeat this entire pathway. Additionally, those particular products from that deiodination can return to the circulation where they may have secondary effects. They may be bound to proteins in the circulation, and binding to proteins can impact the ability of these hormones to diffuse into their targets. And then finally, what's missing from this also is the fact that there are active clearance mechanisms that can even destroy thyroid hormones, remove them through the kidney, through the urine and the bile before they are even active at target tissues. So the complications come from trying to figure out where the most important components of this system are as far as determining what gets to tissues. And the reason this is of interest to fish endocrinologists, the reason we have to go past what's shown in these textbooks, is that I've told you already that it looks like fish are breaking the rules in one way, in that instead of the classic activation coming from the hypothalamus, they appear to have, at least in the goldfish that Dick studied, an inhibition coming from the hypothalamus, suggesting that, in fact, they put the brakes on this system instead of activating. The person who's done the most work and just beautiful work on fish thyroid physiology is Jeff Eales, retired from the University of Manitoba, now living a happy life in a cabin on an island outside of Vancouver, and still writing reviews, I might mention, years after he's retired. Jeff made the argument based on many years of careful research in salmonids, that he worked primarily with rainbow trout, that in fish systems, the availability of hormone to the receptor is primarily regulated by these peripheral processes, not by central processes. And he had a number of different arguments for that. He spent many years doing very careful studies of the expression and the activation and the biological activity of the inner and outer ring deiodinases, and showed that the deiodinases are very dynamically regulated in target tissues in fish. They're regulated by nutritional state, they're regulated by stress, they're regulated by hormones like estrogen and growth hormone. And that regulation appears to be independent of the upper central axis. He also pointed out that there are a number of kind of unintuitive aspects of fish endocrinology compared to mammalian systems. For example, if there are periods of time when T4 increases, when thyroxine increases in the blood, that can be either exogenous thyroxine, you could administer it, or it can be released from the thyroid gland. You'd expect that you'd see an increase, if the simple model is the mammalian model's there, you'd expect to see an increase in circulating T3 if T4 is being driven through deiodination in the outer ring and the T3 was returning to the blood. And in many cases, he's done the supplementation experiments. You supplement with T4, you don't see an increase in T3, suggesting that in fact the target tissues aren't humbly accepting that T4 and using it to activate their receptors, but instead destroying it. He also pointed out that if you increase T3 in the blood, which you might expect to have a negative feedback at the level of the pituitary, T3 doesn't appear to have a negative feedback effect on T4. T4 stays stable in the blood. That would suggest that the only way T4 can have a feedback is to be deiodinated through outer ring deiodinase. Again, putting the focus not so much on the provision of hormone, but on the metabolism of the hormone in target tissues. He also pointed out that T4 and T3 in fish don't change in concert many times. Again, if there was a simple process where you drove T4 into target tissues through the hypothalamic pituitary thyroid axis and then deiodinated it for action, you might expect that the T4 and T3 would change in concert in the blood as the T3 is being produced. That doesn't happen. So he had a number of arguments. I think one of his reviews had 7, 8, or 9 arguments about what was going about the fact that fish appear to have a system that's much more peripherally regulated. And he argued that in fact in fish, the system works through these deiodinases to maintain what he calls a T3 set point. That T3 is the critical hormone that's being produced, and that function of T4 and the entire, in fact, HPT axis, hypothalamic pituitary thyroid axis, is simply to provide enough prohormones so that the target tissues can decide what they want to do with it. They can just destroy it if they choose to, they can use it if they want. And so the HPT axis becomes much less important in fish. The peripheral utilization of that hormone becomes the area of focus. And Jeff did some really beautiful work proving that that was true. Now, Jeff can get away with that because he pretty much had the patience and wisdom to do those kinds of studies in fish. But the problem is when most people get into studying fish thyroid physiology, they discover, especially if they're more familiar with kind of the mammalian system, that studying the fish thyroid is challenging. One thing is fish don't have an encapsulated thyroid gland, such as you'd find in mammals or in reptiles. This happens to be the lower jaw. This is a split open lower jaw. We've removed the ventral aorta from a red drum jaw. And this is a technique that was developed many years ago that deposits silver into thyroid follicles, and then you basically develop them just as you develop an old black and white negative. And everywhere that's dark is where silver is precipitated inside thyroid follicles. And you can see that thyroid tissue is kind of distributed throughout the whole front end of this guy. That really eliminates one of the most powerful surgical techniques that you can use to study thyroid function, which is thyroidectomy. Mammalian studies, the surgical removal of the thyroid really helped elucidate some of the most important functions, because then you could replace the hormone and you could do classic extirpation replacement experiments. You can't do those in fish. The other problem, of course, you have is that actions of the thyroid gland can often be quite cryptic. This is a gland that stores a large content of hormone. And when you try to defeat the function of this gland, it can produce hormone a long period of time. You can't remove it by thyroidectomy. And the various activating and deactivating enzyme pathways that exist in the tissues actually work to defeat your experiments. Compensatory adjustments are very common in this system. So for example, if you administer thyroid hormone to a fish, what they'll do is they will alter the activity of the inner and outer ring deiodinases to actually remove the hormone that you're administering and maintain that constant set point that Jeff Eels has proposed exists in those tissues. So often the animal has systems in tissues that will intentionally defeat the kinds of experimental manipulations you're trying. You look at that diagram on the previous page and it comes back to, well, what do we study? Do you look at the HPT axis? Do you look at TSH or TRH? Are you interested in the hormone in the blood? Are you looking at the receptors? Are you looking at the deiodinases? Again, Jeff Eels was able through his career to do all of those things, and that's why he had the most comprehensive model. But someone coming into this would take a look at that and say, okay, so where do I start? Which endpoint is most important? Other problem, I don't need to tell fish people about this, is the fact that we're dealing with very diverse life histories, habitats, reproductive strategies in fish. And some orders of fish have been separated probably as long as reptiles and mammals have. So there's a lot of opportunity there for divergence and specialization, which can confuse the picture. And then finally, some people think it's boring. Not me. I really feel that there's still fundamental interest questions that remain to be answered. And let me tell you about a few of those. So I've reached a point in my career where I can play the role of kind of the cranky old guy, the old scientist who sits in the back of the room and complains about things. So I've got a few cranky points that I'm going to put into this talk, and you can tell when they're coming up because I've got them here with a picture of Abe Simpson pointlessly yelling at a cloud. And there are a few things that remain unresolved already that are really remarkable. For one thing, Dick discovered that negative control of the thyroid axis back in the 1960s. We still don't know the mechanism of that inhibitory hypothalamic control, which is pretty remarkable to me. And I just mentioned that because when I was sitting in the audience in 2012 at the North American Society for Comparative Endocrinology meeting at Ann Arbor, and Dr. Volkoff was giving a talk about how she had techniques for injecting microliter amounts of bioactive peptides into the hypothalamus of fish, I said to myself, this is how it needs to be done. Here is the person who has the technique that can look at that inhibitory hypothalamic control of the thyroid system. So that's one of the reasons I'm pleased to see that you're moving toward that direction. That's cranky point number one. But do we really need another species? I mean, here in fish endocrinology, we've already got such charismatic species as the long-jawed mudsucker and the climbing perch and the Asian stinging catfish and the barfin flounder. Why have I moved on to another species when goldfish was doing it for us? Well, I don't know if you're familiar with the red drum or not. This is a perciform cyanid fish. It's very common along the coast, the eastern coast of the United States, from Massachusetts to Mexico, really. And it is an extremely popular fish in the recreational fishery. If you can stand on the beach in Port Aransas, throw a bit of the water and an animal will feed you for a week, and the meat is delicious. It is a very popular sport fish all the way from North Carolina all the way around to Mexico. The problem is, of course, that it wasn't exclusively a recreational fish. In the mid-70s, the chef Paul Prudhomme in New Orleans developed a dish called blackened redfish, which became a fad dish in New Orleans and resulted in overfishing of the redfish populations in the Gulf of Mexico. Now, if there's one thing in Texas you don't mess with, it's their recreational fishery. It's a huge industry in Texas, and the recreational fishermen were very concerned because the population of red drum was declining rapidly as they were being fished out by these purse seiners who were taking them to New Orleans and turning them into dinner. And so the recreational fishermen were able to get commercial fishing of red drum outlawed on the Texas coast. And at the same time, the Texas legislature, in their enlightened support of recreational fishery, decided to develop both aquaculture and stocking programs and funded these programs through the Texas Parks and Wildlife Department. And there are now about a half dozen red drum farms in Texas. It's not a big industry, maybe $5 to $7 million a year financial impact. But the Texas Parks and Wildlife Department has 3 hatcheries where they produce anywhere between 20 and 30 million fingerlings a year that they dump into the bays. And it was easy for them to give us a couple hundred fish every so often when we needed them. So we had a supply of animals. And they also were interested in developing them as an aquaculture species. So we had about 10 years of financial support in collaboration with our colleagues in the Department of Wildlife and Fisheries Sciences to learn a little bit more about raising these animals in captivity. Mostly it was diet development, husbandry, but I was able to tag along and do a lot of thyroid work as well. And so it supported our lab quite nicely. So yeah, sure, why not? Why not another species? This is a Perciform cyanid. As I said, it's euryhaline, warm water, fast growing, abundant, available. And so we got started by doing some basic descriptive studies. Again, we have some interesting resources here in Texas. The Dow Chemical Company has a massive chemical plant that's located in Freeport, Texas, down on the coast. It's supposedly the largest chemical complex in North America. And it was built to extract magnesium during World War II from seawater. And so it has huge. And the red drum that are wild in the Gulf of Mexico will come into these canals and feed on those invertebrate populations. So basically what we had is a captive, not captive, they were free ranging, but an easily captured population of wild red drum. And one of the first things we did is we spent a couple of years going down to Dow every month and taking tissue samples, blood samples, morphometric data, everything we could on these animals to learn as much as we could about them in the wild. We got lots of help from the Dow employees, one of whom, of course, was a graduate of our institution. And what we found pretty quickly is that there were substantial levels of both thyroxine and triiodothyronine that were circulating in the blood. There was nice seasonal cycles. And this was a good start because, again, it suggested to us that this is not a boring endocrine system, at least in these wild animals. There are substantial levels. This is a lot higher than the levels I was encountering in goldfish when I was measuring them at the University of Alberta. So we get started. One good thing about this fish, elevated dynamic seasonal levels. This is a fall spawner. This is the summer period. It looks like lots of things are activated here during the summer. Next thing we did is we did some hormone administration studies. We repeated some of those TSH injection studies I'd done in the past, and we were really pleased to see that bovine TSH activated almost immediately the release of thyroxine from the thyroid gland in red drum. This is actually a study we did in collaboration with our colleague Scott Jacques at the Texas Veterinary Medical Diagnostic Laboratory. He actually had an immunoassay for bovine TSH. This is the only study in the non-mammalian vertebrate I know in which we've measured the heterologous TSH in the blood at the same time we've measured the endogenous thyroid hormone response. You can see that when TSH goes up in the circulation, you get an almost immediate increase. So the thyroid is very sensitive to stimulation by TSH. You should also see that, albeit mammalian TSH is cleared out of the blood quite quickly, whereas the resulting thyroid hormone response is much more prolonged. And that really reflects a difference in clearance rates between the glycoprotein hormone TSH and the amino acid hormone T4. So this is great, more good news. We did some feeding experiments and some hormone administration experiments and gave us exactly the opposite result to what we expected. We were looking for anabolic effects of thyroid hormone by incorporating them into diets. This was done in collaboration with Hae-Young Moon, who was a graduate student in wildlife and fisheries. And you can see here with our control fish and our fish in which we give progressively higher doses of T3 incorporated in the diet, that in fact it had exactly the opposite effect. I gave a talk once about these data entitled it, "Honey, I Shrunk the Fish," because essentially these animals burned up their muscle. And this really underscores a critically important aspect of fish thyroid function. You've got to be very careful with your hormone administration. Thyroid hormone administration to happy, healthy, growing animals is be counterproductive because those animals, we were there in our aquaculture facility, they were producing all the endogenous thyroid hormone they needed, and we were making them hyperthyroid with these doses. So for future studies you'll see in the next few minutes, we tried to be very careful about validating a choice of dose when we administer thyroid hormones to these animals, and in fact reducing the period of time that we that we administer the hormone, so that it's not. This was a particular study in which the hormone was included in their diet for 6 weeks. We were really asking for it with that. Reducing the amount of time is important. All right, and then the third thing we got into was photoperiod and diet. Now, one famous effect of diet has been observed in many fish and mammalian species, which is when you remove food, that tends to deactivate the thyroid system. And that's seen as an adaptive response. Thyroid is very active in promoting metabolism. Survival of starvation requires a decrease in metabolic activity, and therefore shutting down the thyroid when you remove food seems to be a very important component, not only in mammalian physiology, but in fish physiology as well. So Lisa Perez was a master's student many years ago, and she was the first one to jump into this, looking at removing food. We call it fasting for 60 hours here in Red Drum. And interesting things were going on with this. When she first did these experiments, we were only taking blood samples at fixed time intervals after the initiation of fasting. And we really didn't find consistent effects. And that didn't make any sense to us because everybody else had reported that this was something that was clearly exhibited when you remove food. Lisa ended up going on to do a little bit more characterization, and she was the first one to show that in fact, choosing these kind of arbitrary times for sampling was not going to be an effective approach because in fact, changes in thyroid hormone levels in the blood in Red Drum were very dynamic in our systems, in our aquatic systems here in our biology building. About this time when Lisa left, Kevin Leiner came along to do a PhD. And we had been talking to people in aquaculture. And one thing that the people in the aquaculture industry told us is that they found it very productive to leave the lights on. They tried to leave the lights on in their facilities as long as possible because the fish grew faster when they were subjected to long photoperiods. And we replicated those experiments in the lab, regardless of whether fish were fed a maximum diet or a 5% body weight diet. On long days, they grew faster over a period of 28 days than they did on short days, suggesting that there's an impact of photoperiod on growth. And we were, of course, a thyroid lab, so our explanation for that as well, it must be impacting thyroid function. Kevin took a chronobiology class from our department head, Dr. Vinny Cassone, while he was here, and came back all excited about the fact that we really needed to incorporate some components of circadian understanding into our study of fish thyroid physiology. And Kevin did some really remarkable experiments that involved great stamina in taking blood samples from animals. In particular, what he started doing is his standard paradigm was to take a 24-hour period, bleed a different tank of animals every 3 hours during that time, and fully characterize changes in circulating thyroid hormone levels throughout a 24-hour period. And he did this on long photoperiods, short photoperiods, different feeding protocols. And the first thing we'll just say is that, in fact, this isn't boring at all. When you start doing these 24-hour samplings, what you discover, regardless of whether animals are on long or short photoperiods, is that T4 in particular exhibits very robust daily cycles in our animals in red drum. T3 is less cyclic. This kind of looks like Jeff Eel's T3 set point. But in fact, the reason that we were having problems in Lisa's experiments is because Lisa was very conscientious. She'd get up in the morning, she'd come in, 9 o'clock, she'd bleed her fish right after the lights went on. And that's the least interesting time of day to actually bleed these animals. Kevin had a different lifestyle. He tended to come in the afternoon, start his experiments then. And what we discovered was that's when the hormone is peaking on a 24-hour basis. And he's done a whole lot of these experiments and shown that this is a really robust cycle that we get in T4. And that became very interesting to us. Again, we do see what Jeff Eels had predicted, the independence of T3 from T4, but I'll come back to that in a second. So we were interested in what was generating this cycle. And again, we went back to looking at food. Because Kevin was well trained as a circadian biologist, he knew that you couldn't just look at a 24-hour period and say that's a circadian rhythm. Could be something that someone was moving some carts down the hallway and within the first 12 hours, and that's why the hormone went up. What you really want to see to prove that something is a true circadian rhythm is you want to put the animals into constant conditions and allow the rhythm to free run. And again, if you're working with rodents, this is easy. You put a micro switch on their running wheel and you go away. And a month later, you come back and you have a complete description of their activity. If you're an endocrinologist, studying a free running rhythm is pretty difficult because you have to take a blood sample in order to get your data. So what Kevin did is he found every fish tank we had. He set up these experiments with 24 fish tanks in the room, and then basically fed the animals at random times so they couldn't entrain to feeding, and then turned the lights off and put them on constant darkness and claimed he could bleed fish in the dark. I have no reason to doubt that. And showed that in fact that T4 rhythm was maintained for as long as we could do it, all right? Again, we're going to run out of fish in fish tanks eventually. So he bled a different tank every 3 hours for 72 hours and showed that sure enough, that rhythm is free running. And it looks like the T3 rhythm the T3 levels are maintained as well. Again, a true circadian biologist would want you to do it for a month, but that's just not practical in endocrinology. What turned out to be more interesting was that when you take food away, the rhythm continues to run, but its amplitude decreases. And the longer you go, the lower the amplitude of the cycle becomes. And we figured that if you kept it going for a while, you would in fact see the cycle of T4 disappear. And interestingly enough, the T3 was declining as well. So what this all means is that Kevin came to the conclusion that this cycle was primarily entrained by light, that it's a true circadian cycle, which would argue for it being generated in the hypothalamus, and that the impact of feeding is primarily on the amplitude of the thyroid hormone cycle and not necessarily on the phase of the cycle. But this is a time, again, when it's worth backing up and evaluating the way in which people do thyroid hormone or other types of endocrine experiments. Often when you read a comparative endocrine paper or even a mammalian endocrine paper, they'll say at the beginning of the method section that in order to avoid the complications of circadian cycles, we sampled our animals at one time of day. And when you think about what Kevin's results show you, it really underscores how that kind of approach can miss some of the most interesting changes, either in amplitude. Let's say you decide to always sample your fish right after you get up in the morning. And as we saw with Kevin's feeding experiments, it had a major impact on amplitude. If you're going to bleed at this time of day, you're going to miss that amplitude impact completely. Likewise, if there's a phase shift that occurs and you bleed at one time of day, you're going to miss that phase shift as well. And especially if you're working, for example, you're administering a treatment that inhibits the thyroid system, you have very little room for seeing that inhibition at a time of day in which the hormones are at their nadir, at their lowest point. So it's really important to identify when during the day you see the biggest difference in hormone production, the greatest magnitude of hormone production, and try and focus your experiments on that period of time. So again, no one ever wants to hear this at meetings, but this requires you to do a lot more work to kind of characterize the baseline levels. And this has even been used as a criticism of those kind of seasonal cycles, the kind of cycles that I told you about, which we all have done at some point or other, it seems, going out into the wild and sampling animals. If you consider the fact that you pick one time of day and then come back at different months of the year to sample those animals at the same location, in February, that time of day can be maybe an hour or two after sunrise. In April, it can be maybe 2 or 3 hours after sunrise. In June, especially as you get farther north in Canada, it can be many hours after sunrise. And if you look across these different samples, it's apparent that there is an increase in the activity of your system during that time, but really all you're seeing is phase shifting. And John Leatherland, who was at the University of Guelph, spent quite a bit of space in one of his reviews criticizing all of the seasonal cycle studies, saying that in fact this artifact could be the explanation for much of what was observed in seasonal cycle studies. That interested us because we had an opportunity to actually do that. Again, here's another cranky point. It's really important, I think, because of these potential effects of circadian changes in hormone secretion, to assure that you understand what the baseline changes are in the activity of your system before you go in there and try to sample it or interfere with it. All right, so actually we thought after we felt sufficiently chastised about our seasonal cycles, that we could actually address this issue of going back and looking at diel changes in semi-natural conditions. I wish we could have done it with red drum. We couldn't, but we did have access to something else. We had access to a population of channel catfish that were kept at our aquaculture center under natural conditions. They were in artificial ponds, but it was natural temperature and lighting cycles. And I had done, when I first got to Texas A&M, a classic seasonal cycle study, measuring thyroid hormones over the different months of the animals that we seined up out of these ponds once a month. And again, pretty nice dynamic seasonal cycle, highest levels in the summer. And the question, addressing the point I just made in the previous slide, is that actually an artifact of shifting levels, phase shifting of a normal diurnal cycle. Tom Loader was a master's student who came in and tested the phase shift hypothesis by basically going out three different times of the year, January, April, and July, when we would have seen major differences in the hormone, and sampling the animals in morning, afternoon, and night at each of those 3 times. And essentially, what we discovered from Tom's work is, yes, sampling time is important. In all 3 of those times, the samples that were taken later in the day always had highest levels, except maybe in January when not much was going on. But in April and July, both for T4 and T3, which I might mention were very well correlated, the highest levels were later in the day. But there was no evidence whatsoever of phase shifting. We really think that this is a legitimate seasonal cycle that we're seeing. But we were fortunate at this time because we were able to extend this a little bit more through a collaboration with Jeff Eels. Jeff agreed to do some of the deiodinase measurements for us. And so one thing is, you see these changes on a seasonal basis, you wonder about what causes them. One of the things that could cause an increase in T3 in July is an increase in outer ring deiodination. So we were able to take hepatic tissue from these animals during these different sampling periods and look at the overall outer ring and inner ring deiodination activity in the liver across those 3 different samples. And interestingly enough, what we discovered is that the outer ring deiodination, the deiodination that produces T3, is really pretty stable across those 3 different samples. But the inner ring deiodination was really quite dynamic. It was much higher in the January period, even accounting for changes in temperature and substrate availability. It's about a 35-fold increase in inner ring deiodination during these cold months of the year, suggesting that maybe inner ring deiodination is actually a critical component of the overall thyroid economy by destroying some of these hormones in the coldest period of the year when the animals are growing slowest. So again, this kind of fit in with Jeff's model of the peripheral deiodinases playing a critical role. So how can the cycle be generated? One way the cycle can be generated is simply via the TSH system. You drive T4 into the blood, and then T4 feeds back to inhibit the production of TSH from the pituitary gland, and your cycle is due to T4 entering and then subsequently deactivating its own production. And that means that TSH could be driving these cycles. Jeff would argue the opposite, which is that the cycles are being driven peripherally by changes in the deiodinase enzymes, which are controlling the utilization of T4 and the production of T3 and the clearance of those molecules out of the blood. And Jeff and I have gone back and forth on this over the years, but I was a TSH lab, so I'm going to take this one on the left. And the issue is, how can we move from studying the overall thyroid hormone transport to a better evaluation of what happens upstairs? Now, Kevin had also discovered a couple of other things which kind of contradicted the Eels model. He did, in fact, find times when increases in T4 were associated with increases in T3. For example, here's a study on long days where he fed different groups of fish either once a day, twice a day, or 3 times a day, same diet. And really, it's very cool. The magnitude of the peak of T4 correlates with the number of feedings. A single feeding gives you the highest peak, and as you give more feedings, the peak is spread out more broadly over time. But whenever T4 gets above around 20 nanograms per mil, we get a correlated peak in T3 in the blood at the same time. And again, that violates one of Jeff's assumptions about the relationship between T4 and T3. The other thing is, Kevin was able to actually inhibit the thyroid system with T3 feedback. But he did that by immersing his animals at the correct time of day, which was late in the day when levels are highest. He stopped the water going into our flow-through systems. He gave a shot of T3 into the water to immerse the animals. They took up the T3 through their gills, we expect, into their blood, and gave us a physiological increase in T3. And associated with that, we saw an inhibition of T4. And so this suggests that, in fact, there is a negative feedback that's functioning. So putting that together, we came up with a hypothesis that the daily T4 cycles are driven by negative feedback on TSH. That T4 increases in the morning due to a TSH-driven thyroid stimulation. That T4 would then decline in the afternoon due to feedback inhibition of TSH. And if that's true, you would expect that TSH should cycle as well. And here is one of my favorite cranky points. After all these years, there is still no way to measure Telios TSH in the blood. There's no Telios TSH immunoassay. I have tried to get money for this many times, a lot of good reviews for grants, but never funding. We've worked with private companies, we've worked with whoever will tell us that they can make TSH, and we still don't have a Telios TSH immunoassay. That would allow us to measure the pituitary hormone levels. So we did the second best thing. We looked at TSH subunit expression. And again, this doesn't tell you that it's actually the protein's being produced and secreted, although this kind of work has been done in mammals to suggest that the expression of the TSH subunits is reflective of how much gets into the blood. Richard Jones was Kevin Liner's student worker back when Kevin was a graduate student. Richard was an undergraduate, cleaned tanks for us, did all the grunt work in the lab, and then went off to medical school. After 2 years in medical school, I got a phone call from Richard saying, I don't really want to be a doctor. I want to come back and finish Kevin's experiments. So Richard dropped out of medical school, came back to do a PhD. And Richard was a cloning fiend. Within a first year, he had already worked up quantitative PCR techniques for evaluating the expression of the subunits of TSH, both the alpha and the beta subunits of TSH, and for deiodinase 2, the outer ring deiodinase, and deiodinase 3, the inner ring deiodinase, and TSH receptor. And he developed techniques that were sufficiently sensitive that he could actually look at expression of these proteins in individual pituitary glands from red drum. So the first thing we did is ask, okay, if this feedback model is working, we should expect to see TSH expression elevated in the morning when it's being pumped into the blood and inhibit it in the afternoon when feedback is working. And that's, in fact, exactly what he found. This dotted line in the back shows you the thyroid hormone cycle that these fish are showing. And this is the beta messenger RNA for TSH is being expressed in the pituitary glands of those animals. And you can see this beautiful reciprocal relationship arguing for the fact that T4 is having negative feedback. And he even had one experiment that didn't work well. He got a weird thyroid hormone cycle, but even with that weird thyroid hormone cycle, he still got the reciprocal changes in TSH beta. So we had interesting information there suggesting that feedback was actually occurring from the blood to the pituitary gland. So that gave us the hypothesis that T4 had to be deiodinated in the afternoon to exert that negative feedback through T3, which is the active hormone. And the deiodinating enzyme, of course, is outer ring deiodinase. So what about outer ring deiodinase? Does it vary as well? Well, the first thing you do whenever you're doing deiodinase studies is you have to characterize the deiodinase function in different tissues. Richard got off to a start here by looking at it in the pituitary gland. And you can see here that deiodinase 2 expression didn't really give us a clear signal. It varied, but not much of a clear signal. But interestingly enough, deiodinase 3, the deactivating deiodinase, showed us this same reciprocal relationship, which is what we would have expected if deiodinase 3 was involved in the negative feedback system. And so this is moving our focus a little bit from deiodinase 2 back to deiodinase 3. This suggests that the feedback of thyroid hormone to the pituitary may not be achieved necessarily by regulating the activating deiodinase in the pituitary, but maybe by regulating the deactivating, destroying the destruction of thyroid hormone by deiodinase 3 in the pituitary. So the way to do that is to see if feedback actually impacts the deiodinases. So Richard expanded on Kevin's immersion studies and developed an immersion technique in static tanks, which allowed him to increase T3 and T4 independently in the blood of fish by immersion in those solutions. And the first thing you do is you always go look at the liver, because the effects of thyroid hormone on deiodinase expression in the liver has been extensively characterized in tilapia and salmonids, and you got exactly what he would have expected. If you immerse our animals in T4, and that's the blue line, you see it doesn't really have much effect on deiodinase 2 expression in the liver, but it has a really dramatic effect on deiodinase 3. And that had been reported before. And that's part of that compensation mechanism I told you about. Essentially, what it means is that when you pump an animal full of thyroid hormone, it actually changes the activation of its deiodinases to reduce the production of thyroid hormone in target tissues such as the liver. And that's going to defeat your ability to impact the liver with the hormone that you've administered. So this was a great validation. This is exactly what you're expecting. So then he went and did the same procedure for the pituitary gland. And he looked at TSH beta. And when he immersed these animals in either T3 or T4, TSH beta, lo and behold, was inhibited over time by both of those hormones, suggesting that there is an effective negative feedback. And even for icing on the cake, looked at the alpha subunit of TSH, which most people don't look at. It was inhibited as well, really nicely. So what we have here is the suggestion that both of these hormones can exert negative feedback. T4, perhaps through deiodination, T3 maybe directly through uptake by these cells. But the most interesting aspect of this to me was the effects on deiodinase 3. When you submerge these animals, you immerse them in these hormones, we see somewhat of an effect on deiodinase 2, but we consistently saw that deiodinase 3 expression was inhibited by feedback of both of these hormones. And again, we'd expected deiodinase 2 to be the action. This is the activating deiodinase. This is the one that's going to convert T4 to T3, which is going to have the negative feedback. We hadn't expected to see such a dramatic effect on the deactivating deiodinase, but what it suggests is that maybe the more dynamic action in regulating thyroid hormone feedback is through regulating its destruction in target tissues instead of its production. So when we submitted this to General Comparative Endocrinology, said, yeah, you got to have a model. So we agonized over this. There's no easy way to kind of put together a model that encompasses inhibiting inhibition. But basically our model is that during the morning, TSH is being driven out of the pituitary to stimulate an increase in circulating T4. As T4 rises, it feeds back to the pituitary gland. It's deiodinated by the existing deiodinase 2 in the pituitary gland. And if allowed, that T3 that's produced inhibits the production of TSH, resulting in a decrease in TSH and a decline in T4 in the afternoon. That decline in T4 releases deiodinase 3 from the inhibition, and that then destroys the T4 feedback and reverses the process. TSH begins to go up and the cycle starts over again. So this is essentially a self-sustaining oscillator that exists in the pituitary gland and could explain the cycles that we're seeing, of course, with the caveat that we still can't measure TSH and don't know that it actually is the hormone that's responsible. So what have we learned from all this? Well, first point is that in mammalian literature, they focus greatly on deiodinase 2. Deiodinase 2 is the activating enzyme. Presumably, you can't have thyroid hormone effects without expression and action of deiodinase 2. But what we showed is deiodinase 3 appears to be much more dynamically and directly regulated, and we think it deserves more respect. And in that regard, we definitely agree with Jeff Eels. He's felt that deiodinase 3 is a major regulator of target tissue response to thyroid hormone, and it really needs to be studied more. I have a former student who works on manatees and elephant seals and is doing deiodinase studies and is doing it all on deiodinase 2, and I keep telling him he needs to do 3, and he is hard to convince. He says, no, we don't see it. It's not important. I think it is, and we need to do more work with it. So in the end, what have we learned from all this? Thyroid hormone dynamics in warm water fish are not boring. I don't think there are any boring endocrine systems, just boring fish species, or fish that have been made boring by being acclimated to the laboratory for many generations. We really want to focus more on TSH. Dynamic cycles of TSH expression suggest an important role of TSH. And one of the big advantages in studying TSH, as I said before, is it has a high rate of clearance from the blood, meaning its changes could be very rapid. Thyroid hormones tend to be dampened in their changes because of course, the protein binding in the blood, different rates of clearance. But if you're interested, for example, in the effects of endocrine disruption on the thyroid system, you may be able to see changes more rapidly if you're examining TSH expression because TSH turns over so quickly. The other thing is that circulating levels of these hormones can inhibit can inhibit TSH at physiological levels. And we think that deiodinase 3 may be an important point of focus, as I said on the previous slide. People should spend a little bit more time looking at it as a way to regulate thyroid hormone availability at receptors. Maybe deiodinase 2 is more constitutively expressed, hormone is activated, but the real action comes from destroying the hormone instead of building it. And therefore, a kind of a punchline here, thyroid hormones may be playing an important role in the promotion of daily cycles of metabolic activation associated with nutrient intake. The question comes back is, well, why have these cycles? Well, these are animals that are diurnally active. They're feeding during the day. They're digesting and assimilating nutrients during the day. Thyroid hormones are going to promote that process. And what you have here is a mechanism for anticipating those daily cycles and firing up processes of nutrient assimilation during the photoperiod, the photophase during the day, so that the animal is already prepared for assimilating that food, those nutrients as they come in. And again, it's so easy to do gene expression studies and biochemical studies, but what I think these kinds of studies underscore is that we really need to know more about hormones, how hormones get to their targets, about how they actually are transported through the blood and are made available, because in many cases, they may not get to targets at all. They may get destroyed. And all of that work that we've done in characterizing the various proteins that are expressed in target tissues may be useless because the hormone's not getting to the receptors. Okay, looks like I've used up pretty much all the time I've got. I just had a couple of last slides. Everyone wants to throw in a couple of surprises. We've done radioactive iodine PET CT imaging of fish, and it suggests that in fact the subpharyngeal thyroid, which is right here, is in fact the most active area. Maybe there's a lot more thyroid hormone that's being produced from the subpharyngeal region than has been suggested. And Allison Wilkes, who did a PhD with me, did that work. And recently we've been getting into looking at sodium iodide symporter. And again, don't give up on some of these exotic fish. They will throw you some really interesting curveballs. We went in to look at the symporter to see where iodine was being transported in these animals. And it was in a lot of predictable places, in the digestive tract in particular, and in the thyroid, but also in the central nervous system, which we hadn't expected. And this was discovered by an undergraduate, Julie Butler, who's now doing a postdoc at Stanford University, finding out that there are some places where some proteins which have been well established in mammals are in fact being utilized in different ways. And Nick Holloway, who had just moved to Berkeley to do a PhD, finished a master's with me last year. And he's shown that in fact, the sodium iodide symporter, which was thought to be exclusively in the digestive tract and in the thyroid gland, is broadly expressed in the brain of zebrafish and in developing follicles in the ovary of zebrafish as well. So and broadly expressed in the brain across different fish species, including channel catfish, hybrid striped bass, tilapia. So we're learning some things from red drum which are fascinating and unexpected, and that to me is the real joy of working with fish. They tried just about every experiment with their endocrine systems you could think of, and that's what makes them fun, is they keep throwing those surprises at you. So in conclusion, I'd like to thank all the people in my lab. These are all graduate students, my current graduate student, Bree Meyer, who gave us the idea of looking in the ovaries for the NIS, master's students and some undergraduates, Bill Cohn, who's hung around our lab for 25 years, has been a great contributor to the molecular studies. I'd like to thank my collaborators in wildlife and fishery sciences, especially Dr. Delbert Gatlin, who provided the fish and worked with us on all those nutritional studies. Scott Jacques, who helped us with the various studies related to the TSH and the sodium iodide symporter, the PET CT scanning, and of course, the Dow Chemical Company and Texas Parks and Wildlife, who provided us with animals. And especially Pete Thomas has helped us out. Jeff Eels at the University of Manitoba has been a fantastic colleague over the years and is a true intellectual of fish thyroid physiology. And then State of Texas has paid for much of this work over the years. And with that, I'm happy to go ahead and move on and take any questions and show you some pictures of people taking blood samples from fish in various locations in Edmonton, in Mexico, in ponds and in laboratories. And thank you very much for your attention, and I'm happy to answer any questions you might have.
All right, fantastic. Thank you very much for the talk, Duncan. This was certainly a good one to end off with. Very interesting. So yeah, if you do have a question, yeah. So yeah, if you do have a question, please just indicate so in the chat, and I'll call everyone out in order. All right, go ahead, Ed.
That's the equivalent of raising a hand.
Yeah.
Hello, I'm Ann Story, a retired faculty member in psychology, studying reproductive hormones and behavior.
Yeah.
And I really enjoyed your talk, but it was also a great trip down memory lane. I took animal physiology from Jeff Eels in 1970.
Oh, you're so fortunate.
It was great. It got me started, and now I'm, retired. And also friends with Larry Krim for 40 years. So.
Ah, great.
Anyway, thank you.
Yeah, Jeff got many people started. I mean, when you consider the people like John Chang, Glenn Vanderkraak, who are kind of some of the leaders in comparative endocrinology now, started out as master students in Jeff's lab. He inspired a lot of people.
Yeah.
And he's still going, as far as I tell. He's got a lot of data that he's still writing up.
That's amazing, because I was a third-year undergraduate when I took the course. Wow. And I'm retired, so.
All right, go ahead, Cole.
Yeah, thanks so much. I really enjoyed that. I was laughing out loud at some of your anecdotes you're bringing up, especially with Aubrey Gorman. That's pretty funny. Um, yeah, I was reading this old paper by I think Martin Sage back in the 70s, and he
Oh
was
yeah.
bringing up all these. He's like thyroid hormones have been studied and all of these things, and I think people are like kind of getting bored with it because we like know all we know, like we can know. Um, but I don't know. I mean, like how other than you, I feel like not a lot of other there's not a lot of fish thyroid endocrinologists out there anymore. And I mean, what kind of arguments can we make to keep I don't know persuading people to give us money to study it?
That's a really interesting point, because I was thinking as I was putting this together, when you talk to graduate students, a lot of times the questions you get is, well, this is a really interesting area. Where should I go to do a PhD in this area? And you look at the number of kind of dedicated fish thyroid laboratories, and there really aren't that many. And I firmly believe that is an outcome of this prejudice against the ability to do quick and dirty science with the thyroid gland. So I talked about Paul Licht. For 30 years, Paul Licht had what I would consider to be the most successful comparative endocrine program in the US, because he was solidly funded by the National Science Foundation to do work on gonadotropins and on reproduction. And in the process of that, we found some really interesting things about the thyroid. And toward the end of Paul's career, he made the transition from working on reproduction to the thyroid. And I remember Paul telling me that was when he started having trouble getting funding. It's hard to convince people that the thyroid is critically important. I mean, everybody starts their reviews by saying, well, the thyroid does all these really important things. But it's hard to study. It's hard to do these kinds of experiments where you can get immediate and unambiguous results. And honestly, I really feel like that's an opportunity that's being missed related to TSH. I think that could happen with TSH if we were able to get the money to develop the immunoassays or some other way to measure it. But kind of going back to your question, when Jeff retired, that to me kind of closed down the best thyroid endocrinology lab in the world, honestly. And then John Letherland retired, and their students are still working in areas that are related to thyroid function, but in many cases, when you look at people like Vanderkraak and John Chang, um, they've moved back toward reproduction, and they've incorporated the thyroid issues into reproduction. Now, somebody who is taking off with thyroid function right now is Hamid Habibi in Calgary. And he's got some really nice analytical techniques for looking at different thyroid hormone receptors. But again, it needs to be, I think, a little bit more comprehensive as far as looking at the entire axis instead of just the regulation of the receptors. It's the kind of thing where you need to find a productive program and then infect it with thyroid physiology, because as long as we're going to keep writing reviews, like your review starts out with the thyroid does very important things, and there's no question that it does. It just does them in a fairly cryptic way, and finding the right types of endpoints to look at, I think, going to be the critical way of moving forward in the future. And I really do feel like the deiodinases are something that the thing Jeff had going for him that the rest of us didn't is Jeff could look at the expression like the rest of us, but Jeff had a really beautiful system for evaluating the biological activity of the deiodinases. He incubated the tissue homogenates with radioactive hormone and then did HPLC separation of the products. So, I don't see anyone doing that kind of technique now. So I am discouraged, honestly, about the future of fish thyroid physiology. And it may be that the best way to move forward is to focus on the more practical sides relating to endocrine disruption. And again, that's something that I think is still somewhat of an easier sell than just the thyroid is interesting. Let's study it. I don't know, that's not really an answer to your question. That's a little
No, yeah.
bit.
And that's a direction I see like in the literature as a endocrine disruption for sure. But it just blows my mind. We still don't know what thyrotropic factors out that, you know, it's like, because you're proposing the thyrotropin inhibitory factor, and like there's no consensus, and yeah, there's still so much to learn in fish.
Yeah, and it could be different from one species to the next, unfortunately.
Sure.
You gotta find the animal that's not boring.
Yeah.
I see Dr. Volkoff there nodding her head. I mean, she really has taken the hypothalamus of the goldfish and turned it into a fantastic system for manipulating the neuropeptide content. And I still feel, I don't mean to put you on the spot, Ellen, but I do feel like you're in a great position to do a little bit more thyroid work with those.
Well, that was our next step with Cole. Like we were going to take the hypothalamus and do ICVs with thyroxine and do cell culture with the hypothalamus. But unfortunately, with that COVID thing, everything is so complicated. But that's my next step.
Okay. Because, you know, essentially all I've been doing all this time is using thyroxine as a bioassay for TSH. And, you know, the only person who's actually been able to measure TSH is Bob Denver did a lot of it with a TSH immunoassay when he was in Paul Lick's lab. And that's where he discovered that CRH is potentially the TSH releasing factor in non-mammalian tetrapods. So, you know, having that TSH immunoassay, I think would really make a big difference. I was lucky when I was working with Dick because we had a really dramatic result. And I think that's the best thing that you've got going for you in the goldfish. We know that when you lesion the medial basal hypothalamus, it was I forget the various locations, I'm sure you know them better than I do these days. When we did those lesions, we got a response within 4 or 5 hours. And that has to be a TSH mediated response that we saw. But again, the thyroid hormone levels stayed up. They stayed elevated for a prolonged period of time. And that didn't tell us anything about kind of the dynamics of the TSH production, because as I showed you, you give them a shot of TSH, thyroid hormone stays elevated for hours and hours longer than the TSH does. So, you know, using that technique, you've got a way to turn on TSH production. The question is, can you then go in and shut it back down again with some kind of inhibitory, you know, administration? I don't know if the timing has to be really critical. I'm sure.
Well, something that was great in the presentation, you showed us that if you do that in the morning or in the afternoon, you might have different results. So you actually should have to do that at least 3 times a day.
Yeah.
So that's a lesson learned.
And no one wants to hear that.
Yeah, because it's a lot more work.
Yeah, exactly. I've had a couple of students come in and do TSH injections at different times of day to see if the sensitivity of the thyroid differs between morning and evening. And we never really found that to change, at least not so we found a significant difference. But I still feel like it should be fairly straightforward once you find the right time of day to turn the system on. The problem that's more challenging is turning it off. If you're looking for an inhibitory factor, you've got to find the time of day when the system is most actively activated and show that you can prevent that. Just as Kevin did, the really clever thing Kevin did with that T3 inhibition experiment is he didn't just soak them in T3 for the whole day. He waited to just the time of day when he thought that the TSH was being activated, and that's when he administered the T3. So you're right, once you have that time of day, then you can focus on looking at inhibition. But the problem is, when I was working with goldfish, the thyroid hormone levels always seemed so low in the animals that we had in the systems in Edmonton that it was going to be difficult to look at inhibition of those levels. It was going to be much easier to look at activation.
Did you look at just one reproductive stage, or because it might vary with reproductive stage as well?
Yeah, so Tracy Marchant and I did a big study that never got published, where she was looking at growth hormone and I was looking at thyroid hormones. at that time, there was in the literature a couple of publications by Richard Spieler from LSU, who looked at daily cycles of thyroid hormones in goldfish and showed that they were pretty dramatic daily cycles. So we were expecting that. And so we looked at, I think I just did lights on to lights off. I didn't do during the dark because I don't think they let me down in the aquatic facility in the dark in Edmonton for some reason. It was kind of creepy down there in the dark anyway. But we looked at, regressed fish, recrudescent fish and mature fish, looking at thyroid hormone cycles. And we found them. The magnitude just wasn't very impressive. And now I feel like diet has a major impact on that. Again, getting back to what we saw with the red drum, when they're in great shape, they're on an ideal diet and they're growing fast, they're producing all the hormone they need. I'm not sure that the diets we were giving them in Edmonton at the time were necessarily ideal for what they had. And maybe your fish are you're a nutrition person, maybe your fish are a lot happier and healthier.
And they're fat. They're fat.
Yeah, and they'll have much higher hormone levels. And once you find that peak hormone level, that's where you're going to be able to shut them down. So yeah, we did it at different times, and we did see some increase in magnitude, but it was also associated with changes in temperature. And
very simple.
Yeah, exactly.
Any more questions from folks? Sean, I have one quick one. I guess it has two parts. So if the answer to the first question is no, then I just won't ask the second part. But this circadian rhythm in thyroid hormone levels, do you know, is that like common among all vertebrates, or is that just in fish?
I would like to think that it is, yeah. Well, so what we see in mammals, when you look, and again, we've got a lot of data on humans and animals in the veterinary trade. We don't see as dramatic a circadian cycle, and that's because the binding of thyroxine in the circulation is quite different in mammals. There is a high affinity protein in mammals that doesn't exist in fish that functions to really soak up a huge amount of thyroid hormone. So again, in fish, we're dealing with circulating levels between 5 and 15 nanograms per mil. In humans, they're between 80 and 100 nanograms per mil, and they're relatively stable because there's a very high capacity for binding in the blood associated with this high affinity protein. Jeff Eel's most recent review a year and a half ago is a really fascinating discussion of this. The review is specifically contrasting human and fish thyroid economy, and he makes a very valid point that you really can't use these mammalian models for establishing baseline function in fish, because the mammalian system is so different in the way in which thyroid hormone is transported in the blood. The presence of this protein tends to dampen out daily cycles. But the way you know they're happening is by looking at TSH. You can see circadian changes in the pituitary hormone, meaning that maintaining this kind of standard saturation of thyroid hormone in the blood of humans is dependent on an underlying circadian drive from the hypothalamus, which pushes the hormone into the circulation. So it depends on the literature. You can find literature say, oh, there's no circadian changes in the thyroid function in humans because T4 is stable during the day and T3 is very low. But if you look deep in the literature, you'll see that, yeah, there's evidence that, in fact, from the hypothalamic pituitary standpoint, that cycle is there. And I would say that it certainly, given the changes in metabolic status in an animal between the photophase and the scotophase, I would say that you'd expect to see it in any animal which has kind of dramatic daily changes in metabolism. And that should certainly be true of mammals.
Super neat, because I was like, I study mammals, and I'm not an endocrinologist, but I'm doing some stuff with hormones, and I'm just thinking, like I study elk, and they're like a crepuscular animal. So I'm thinking about if it's like, if the advantage is to promote feeding during like I guess during the day in fish, like should I expect then to find different, I guess, peaks at, in the evening and in the morning?
But I think you're going to find it to be much more dampened out. I think you're going to find some dramatic seasonal changes. But I don't think you're going to find, just from the simple mechanism, the simple measurement that we have of looking at total hormone in the blood, I don't think you're going to find very dynamic changes on a daily basis. There might be underlying changes in the activation of the system, but you won't see it because of the way it's dampened by the transport hormones. But there has been work done on a number of mammalian species, and even some work on ground squirrels as well, looking at hibernation, suggesting
Cool.
pretty dramatic seasonal changes.
I guess that's kind of good news for me, because I'm hoping that I don't have to deal with too much daily fluctuation. But
Yeah, well, you still got to characterize it first, all right?
yeah.
You got to experiment once. And again, when people have that, I've stopped getting papers to review for some reason. When people have that statement in their paper saying, well, we took blood samples from one time a day to preclude any circadian effects, my response is usually, prove that to me. I think that you may potentially be missing some really interesting changes by only taking samples at one time a day. If nothing else, at least you haven't identified the most interesting time of day in which you might see a more dramatic effect of your treatment.
Cool. Very interesting. Thank you.
Sure.
Good. Do we have any more questions from folks?
We all set?
If not, well, thank you again, Duncan, for joining us.
Yeah,
This was
I really appreciate
really
it.
enjoyable.
This was fun. Again, it's a real honor to speak to this group and even to speak to a former student of Jeff Eel's as an undergraduate. I was expecting that. So thank you very much for the opportunity.
Thank you again.
Thank you, Duncan.
Thank you.
Bye, everyone.
Stay warm.